Hi dear students, welcome to this online class on waxes and phospholipids. In the module which we started in the previous online class, that's about lipids, steroids, vitamins and hormones, we started the discussion of lipids. In the previous video, we have learned about the classification of lipids into various types, mainly into three: simple lipids, compound or complex lipids, and derived lipids. And in simple lipid, it is again of two types, that is triglycerides, which include fats and oils, and then waxes. And in the derived lipids, in the compound or complex lipids, there are different types of lipids in which a lipid molecule is connected to other biomolecules like protein, carbohydrate, phosphate groups, etc. And in the third category, that is derived lipids, we have so many types which include especially steroids, similarly hormones, some vitamins, etc. So in the rest of the module, we are dealing in detail about a few selected classes of lipids. In today's video, we are discussing two of them. One is, in the previous class, we have discussed in detail about the first class, that is triglycerides or fats and oils in detail. Today we have waxes and phospholipids in detail. Okay, listen. What are waxes? Compared to fats and oils, or which are called triglycerides, which is a triester, that means there are three ester groups, waxes are monoesters. Right? So general formula is RCOOR. So naturally it has two parts. This is the carboxylic acid part and this is the alcohol part. So it is a monoester. Okay? And what about these R groups? Both R groups are long carbon chain. So we can call this is a fatty acid part and this is a fatty alcohol. That means wax on hydrolysis will give two products. What will be there? There will be fatty acid as well as fatty alcohol. Now, we discussed three specific examples for waxes. Number one, beeswax, which is produced by bees, and that they use for making their honeycombs. So beeswax can be obtained from honeycombs. And what is beeswax? Depending upon this R groups, you can see it is myrcene palmitate. That means acid part is palmitic acid. You can see C15. H31 means it is a saturated hydrocarbon with 15 carbons, CH3, CH2, CH2, etc. COO, that's the acid group. And the alcohol part is, this is C30H31. So this is called myrcene palmitate. And then next one, number two, carnauba wax, which is obtained from a kind of plant, carnauba plant. It's a palm tree. It's a Brazilian palm tree, and its formula is myrcene cerotate. So this part is the same. Myrcene group is same. Alcohol part remains the same. Myrcene, this C25H51 COO. This is called cerotate. So myrcene cerotate. And third one, spermaceti wax or spermaceti wax, which is obtained from sperm whale, a kind of toothed whale, and it is this name is chemically it is cetyl palmitate. Again, this compared to this beeswax, this part is same. It is palmitic acid or palmitate, and this part is cetyl palmitate. So these are waxes. So any waxes on hydrolysis will give two products, fatty acid and fatty alcohol. Now coming to the importance or uses of wax or functions of wax. Generally, we know that waxes act as a protective covering for animals and plants. Especially in plants, there will be a wax coating on leaf, fruits, stem, etc. And that is for two reasons. One, to prevent loss of water, and also it protect plant from the damage of pest. So it's a protective coating. And again, waxes on skin, fur, feather, etc. of animals provide waterproofing, a waterproof coating for them. On feather, skin, etc. there is wax coating, and that's a waterproofing. And it again prevent water loss, and it acts as a waterproofing for animals. Now coming to these specific waxes obtained from various sources, they find use in many fields. Like beeswax are used for making candles, in making shoe polish, and wax paper, etc. Carnauba wax is used to make protective coating for furniture, car, floor, etc. And this spermaceti wax is used in making candles, ointments, cosmetics, etc. Like that, waxes have many applications. Now coming to the next one, phospholipids, which come under which category? Compound or complex lipids. Now what are phospholipids? It is basically a triglyceride, just like oils and fats which we discussed in the previous video. It's a triglyceride, that means the alcohol part is glycerol. So it is a triester, similar to oils and fats. But the difference is, the first two groups are fatty acids. Okay, this acid is fatty acid, but the third one is not fatty acid, it is a phosphate group. So that is the first difference. Phospholipid contain two fatty acid group and one phosphate which is connected to glycerol. So it's a triglyceride with two fatty acid and one phosphate group. So it has three parts. Now number two is that this phosphate group itself has two ester linkage. That means this phosphoric acid, it has OH on the three sides. OH is connecting with this OH, so it's a kind of ester linkage, or because it's phosphoric acid, it's an acid, so and this is alcohol, so it's an ester. To this side also, this R dash, what about R dash? In phospholipids, this R dash is important, and that make it so characteristic. R double dash. R and R dash is just simply, it is a fatty acid like palmitic acid, stearic acid, oleic acid, etc. What about R double dash? It is amino alcohol. Amino alcohols like ethanolamine. Look at this. Ethanol. This is ethanol and this amino ethanol, right? So there are two groups, OH and NH2. So this OH also will connect to OH here of this phosphoric acid group and forming an ester linkage. So actually this is a phosphodiester linkage here, because phosphorus has one oxide to this side and one oxide to that side. So R double dash can be either ethanolamine group. So that if I write it here, if I show it here, it will be like this. That means if this is connected, it will be O, CH2, CH2, NH2, and most probably this gets protonated in this presence of this acid, and it might be NH3 plus appear as a polar group here. So this, if it is ethanolamine, if R dash is ethanolamine, then this phospholipid is called cephalin. So cephalin is a phospholipid in which R double dash group is ethanolamine. If it is another amino alcohol, see this R dash is an amino alcohol generally. Okay, if it is choline, what is choline? You can see, just like ethanolamine, the difference is that N is methylated. N-methylated. Okay, N-methyl ethanolamine it is. So, if it is choline, then it is called lecithin. Lecithin is specifically mentioned in our syllabus. So, what's the structure of lecithin if asked? You have to do it, you have to write it like this. Lecithin is here O, CH2, CH2, N, CH3 thrice, CH3 thrice plus. This is lecithin. And what about this fatty acid group? It can be palmitic acid, stearic acid, etc. For example, if we show it here, palmitic acid, it will be now, it is C15, H31, C15, H31. So this is a specific example for a kind of lecithin which contain palmitic acid as the fatty acid group. Okay, so that's the structure of phosphoric acid. That's the structure of phospholipids. Is it clear? Now coming to the importance or biological functions of phospholipids. It's very important. We have already mentioned about the biological function of phospholipid, that they have important function in forming the cell membrane. That's very important. So how phospholipid does the role of acting as a strong cell wall, and they have actually two important functions. One is that they have emulsifying property. Number one, they have emulsifying property. They act as emulsifiers. Emulsifying property, just like soap. So this, and the second important property is that they have membrane forming property. So these two properties make phospholipids worthy. Membrane forming. So they are membrane forming and they are emulsifying. These two properties of phospholipid make them useful to be part of cell membrane. How does it happen, or what make them useful for this? See, phospholipid can be conveniently represented like this. That is, see, this is a polar group, right? This is a hydrophilic group because it is polar. This phosphate group is polar, it has charges. Whereas this C15, that is a part of the fatty acid, it is long chain, very long, 15 carbons. So I can show phosphate group like this. This is the polar head and it has two tails. See, this is just turned and this makes up the polar head, and it has this two tails. So this is one phospholipid molecule. And let us note it here, this is polar head, okay, which is hydrophilic, and this is hydrophobic tail, non-polar tail, non-polar, okay, right? So that is the speciality of phospholipid. They have got polar head as well as non-polar tail. This polar head is due to the phosphate group, and this polar non-polar tail is due to the fatty acids. Okay, so due to this dipolar nature, or due to this nature, phospholipids can form what is known as lipid bilayer. They are forming lipid bilayer. See, it is like this. In the cell wall, this is how phospholipids make up the cell wall, like this. This is known as, so this is a cell wall, okay. Cell wall is actually having what is known as lipid bilayer. It's of two layers, bilayer. See? So how it is formed? what happens is that the polar head is directed outward and the non-polar tail is directed inward and they are kept parallel. So this is non-polar region and this is polar head, okay, projected outward. Like that, another layer comes, the second layer just opposite, the tail inward, okay, and the head outward like this, such that the whole within the cell wall you have the non-polar groups, and at the surface of the membrane both sides you have the polar phosphate groups. So what is that doing? So these polar groups can see in the cell. So in the cell, if the cell wall is this, having the polar heads on both sides, inside the cell these polar groups can interact with the cytoplasm, which is of course hydrophilic, because aqueous medium, cytoplasm, so this can interact. And also outside the cell also, since there are polar groups, they can interact. So the point is that this lipid bilayer can interact with their polar hydrophilic groups both inside the cell and outside the cell. Okay, and within this hydrophobic non-polar region, protein molecules may get embedded or they may be attached based on the functions. And also this cell wall has a lot of importance or a lot of functions. So what are the functions of this bilayer or these phospholipids? What are the important functions of phospholipids? Number one, as I said, they make up the cell wall. They give strength to the cell wall. They give mechanical strength and structure to the cell wall. Okay, and protect the cell by forming this very strong cell wall. Then they maintain the shape of the cell wall. Another important thing is that they have what is known as selective transport. They allow selective transport. Selective transport. That means cell wall allows some substances into the cell and prevent others. That's called selective transport. That is possible because of this dipolar nature or because of this bilayer structure. So selective transport is important function of phospholipid or the cell membrane. Another thing is what is called cell-to-cell recognition. Cell-to-cell recognition is possible, or actually it is required in many cases. For example, if some enzymes are acting or some hormones are acting, the cell has to recognize the enzyme and the hormones, for which there will be some receptors attached to the cell wall. So this cell wall is helping for cell-to-cell recognition with the help of receptors attached to the cell membrane, and that is required for many enzymes and hormones. And finally, they act as detergents or emulsifying agents to emulsify fat for transport. So for fat transport in the body, phospholipids are helping out, because phospholipids can act as emulsifying agent. Actually, in a sense, phospholipids are similar to soap molecules, isn't it? Soap is also having this feature of dipolar nature, polar head and non-polar tail. Okay, and because of that, this phospholipids, similar to soap, is also having emulsifying property. So that make the transport of fat possible in the body due to this phospholipids. So these are various important biological functions of phospholipids. Thank you.